Okay, well then we will get going here, talking about blood vessels, if I can get the screen to move here. Okay, so title of our lecture here is Blood Vessels and Hemodynamics, which is a term that refers to different factors affecting blood flow. So hemodynamics, hemo or heme always refers to regards to blood, and the dynamics aspect of that word refers to like factors affecting blood flow. So, thinking back to our lecture on the heart, which hopefully everybody had an opportunity to watch, we learned that, the flow of blood, and we learned about this in our lymphatic lecture, it relates to pressure gradients. So we're going to continue talking about pressure gradients here today, how blood flows from high pressure to low pressure, but we're also going to be talking about other things besides blood pressure, like the speed at which blood flows, what we call velocity of blood flow. We'll talk about resistance to blood flow. We'll be talking about a lot of things in this lecture, a lot of different factors. First thing I want to do is just kind of introduce blood vessels a little bit to you. Not a first time introduction, because we talked a little bit about blood vessels associated with our hearts, like the aorta or the vena cavas. So, reading through, blood vessels contribute to homeostasis by providing the structure for the flow of blood to and from our heart and the exchange of nutrients and waste in our tissue. That last little bit there on the first sentence, exchange of nutrients and waste in tissue, is really the entire goal of our cardiovascular system in a nutshell. The reason that we're pumping blood is predominantly or primarily done in order to give our tissues the nutrients that they need, like the oxygen that we pick up in our lungs, the glucose that we pick up from our digestive tract, amino acids, water, other nutrients. Because remember, body cells typically can't walk around and get their own nutrients. And body cells also can't walk to the places where they need to get rid of their wastes. So wastes are also carried by way of our blood in our blood vessels to places like our kidneys to filter out that waste, or in the case of carbon dioxide, to our lungs to be eventually pulled out and exhaled from our body altogether. Blood vessels also play a very important role in adjusting the velocity, which refers to the speed of blood. But what do I mean when I say volume of blood flow? Anybody know what volume refers to? Anybody taking chemistry and learning about volumes in that class? Volume refers to the amount of blood that we have. Thinking back to our last unit when we talked about blood, we learned that about half of our blood is what? Water. Okay. So by adjusting the amount of water that we have in our blood, we affect how much blood we have as a whole. And by adjusting blood volume, we also adjust things like blood pressure, which we'll be getting into that here a lot in this lecture. 5 major types of blood vessels are found in our arteries and really, or in our body. I kind of really classify them as 3 major blood vessels: arteries, capillaries, and veins. Okay, but the book also goes with arterioles, which are actually a form of arteries. Arteries carry blood away from our heart. So they're both arteries and arterioles, efferent vessels, carrying what type of blood? Oxygenated or deoxygenated? Oxygenated. Okay, unless you're talking about the pulmonary arteries. Okay. Now, arterioles are just the smaller arteries that branch from larger and medium-sized arteries, and we classify them kind of independently, or your book does, because they have a big role in maintaining blood pressure. And we'll be talking about them as what we call resistance vessels. Capillaries are really kind of where the magic happens, as I like to say, where the exchange of nutrients and waste takes place. Looking at the figure, you can see they're the smallest. They're also the thinnest as far as their wall goes, which is great because it's easy for things to pass through them. Okay, so exchange is very, very prominent in our capillaries. Venules are the small veins that collect blood from our capillaries and then deliver it to our larger veins, medium, and then eventually large size veins, which are the afferent vessels, afferent vessels carrying, typically anyway, deoxygenated blood back to our heart, where we can then, pump it out to our lungs to pick up that oxygen and start the whole process over again. A little bit of anatomy on the structure of our heart wall, or excuse me, not heart, because I just talked about heart and anatomy. Our vessel walls. We're specifically really kind of focusing on this page about arteries and veins, which both are made up of 3 layers or 3 tunics. A tunica intima, which is the innermost, also known as a tunica interna, which is going to line the inside of our vessels and is exposed to our blood, made up of simple squamous epithelium we call endothelium, similar to the inner lining of our heart, what we call endocardium. And that overlies a basement membrane of loose connective tissue shown in our 2 pictures at the top there. That's colored yellow. At the bottom, we show an illustration of a capillary, and I mentioned on the previous page, are very thin-walled. In fact, capillaries, really, that's all they have, is that tunica interna, the endothelium, and the basement membrane surrounding it. Extremely, extremely, extremely thin-walled, because it's easy for things to pass through, which is what we want to take place with our capillaries. As I said, that's where exchange is happening in our capillaries. We actually call that capillary exchange. Anybody notice anything different when looking at the artery and vein picture here with regards to the tunica interna? Just that innermost lining. So you see in fold what we call valves. Is that what you're looking at? Valves, you can see, are prominent in veins, but we don't have valves in arteries or capillaries. And valves are going to help promote the return of blood by way of our veins, a hemodynamic factor called venous return that we'll be getting into. Look at the size differential of the lumen between arteries and veins. Arteries are a lot thicker walled, which not so much has to do with the tunica interna, but really more so the media and the externa. And since arteries are much thicker walled, look at the size of their lumen compared to the lumen of a vein. Veins are actually more open and actually can hold more blood than our arteries, which is going to affect what we call venous capacitance. Now, as I mentioned, a lot of that has to do with the middle and the outer layer. The media is the middle layer, usually is the thickest, especially we see that with arteries, consists primarily of smooth muscle, and the primary function or principal function of our tunica media is to mediate what we call vascular tone by way of what's called vasoconstriction and vasodilation, where we can either contract those muscles and thus like kink a garden hose when we want to shut down blood flow, or dilate them, meaning that the muscles relax and the lumen opens up more. Now the terms vasoconstriction and vasodilation refer to really arteries, because arteries perform a lot more contraction and dilation, or constriction dilation, excuse me. But we can perform what's called venoconstriction and venodilation, which is specific to veins. So we will talk about venoconstriction. It's just not as effective because there's not as much muscle in veins as there are in arteries. Now, vasoconstriction is something that occurs in response to sympathetic stimulation, which we know is that fight or flight response. Vasodilation occurs in response to decreased sympathetic stimulation or the presence of certain chemicals. NO, we learned previously, is nitric oxide, hydrogen ions, lactic acid. Those are all chemicals that will cause blood flow to increase because of the vasodilation. As far as the outer layer, the tunica externa, really not much physiologically important that I want to talk about. other than that, it really is just kind of there to help hold the blood vessels in place. Okay, let's get to our first hemodynamic factor. So really the kind of first physiologically thing that I want to talk about, what we call venous capacitance. And I actually just used that term on the previous page. Obviously, venous capacitance has nothing to do with arteries or capillaries. It has everything to do with veins and their ability to hold a lot of blood. When I say hold a lot of blood, it's not like they're holding it in a stagnant state. Remember, don't think of fluids in our body like a pond. Our blood especially is like a river traveling through our various blood vessels. But by having a relatively small amount of smooth muscle in our veins, it allows them to kind of just hold blood, not completely stagnant, but kind of accumulating it without changing blood pressure. We call this property capacitance or venous capacitance, like it's listed at the top of the page, which enables veins to act as a reservoir of blood, where at rest, like you guys are all in a state of right now, about 64% of our blood is found in our systemic veins and venules, okay, which is illustrated with our nice pie chart here at the bottom. But systemic veins and venules can perform that venoconstriction thing that I talk about, which is something that they can do to help quickly divert blood if we need it. Venoconstriction is constriction of the veins, which is going to reduce the volume of blood in our reservoirs. It allows blood to go to the needed tissues, like during a fight or flight response, or skeletal muscle, because that's what we use to fight or flight. In other words, to exercise. And this is done in response to sympathetic stimulation from the cardiovascular center in our brainstem, which is actually specifically in our medulla oblongata, that we actually talked about at the end of our last lecture when we talked about heart rate and stroke volume and regulating that. Okay, our next hemodynamic factor I want to talk about is called capillary exchange, which I've already mentioned that term as well. Obviously has nothing to do with arteries or veins and everything to do with capillaries. And this is, as I told you earlier, the whole reason we are pumping blood, which is why it states there is the mission of our entire cardiovascular system, keeping blood to flow through our capillaries to allow capillary exchange. Capillary exchange is defined as the movement of substances between our blood and our interstitial fluid. So it's either the leaving of substances from the blood, we've talked a lot about like how water does that, or stuff entering the blood from our interstitial space, which we've also talked about water doing that as well. But it's not just water, it's oxygen, it's carbon dioxide, it's glucose, it's amino acids, and many, 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 many other things. Our various substances that we move in and out of the blood move through our capillary walls using three basic mechanisms. The most important method, I would highlight that phrase there, is just a process we've talked a lot about, especially at the beginning of the semester, diffusion, where substances move down their concentration gradient from high to low, like oxygen and nutrients from our blood to interstitial fluid, and then into our body cells. Carbon dioxide and waste going from our body cells into the interstitial fluid and then into our blood in the opposite direction that the nutrients and oxygen are going. Now, the way that we move stuff by way of diffusion can be through fenestrations, which are little tiny pores that are found between or found within cells, because some cells that form our capillary wall are fenestrated. I'll show you a picture from my anatomy lecture of some fenestrated capillaries. Actually, I can just do it like this. I'm going to get it downloaded. Some of our capillaries have holes in them. Tiny little fenestrations. Think of these like a strainer that, I made spaghetti last night. You dump your noodles and water in when you want to drain your pasta. Okay, the water is easily going to pass through these type. We don't have a lot of these in a lot of places, but we do have them in places like our small intestine and our kidneys where we're moving a lot of stuff, either into the blood or out of the blood. Most of our capillaries look like this, where they are not wholly, not a bunch of holes or fenestrations in them, but there are these little clefts, little gaps that exist between the endothelial cells. In fact, this is an example of one of those where we see those intercellular clefts, little tiny holes for stuff to pass through. But we also can just do stuff or do movement by just moving the stuff through the cells altogether. And which means that they actually have to go through the plasma membrane into the cell and then out the other side through its plasma membrane on that other side. For our large stuff that we must diffuse in and out of the blood, like our actual blood cells that we learned are made in our red bone marrow, as well as our proteins that we make in places like our liver, we cannot get them in or out of the blood except by way of sinusoids, which are pictured down at the very bottom of this page. And that's what they look like. I always say they look like Swiss cheese. They have these huge holes big enough to allow blood cells, proteins, and other big stuff in and out of the blood. So that's how, for example, our blood cells enter our circulation after we make them in our red bone marrow. We talked about how, for example, we need to remove worn out red blood cells. This is how they're removed from our blood in places like our spleen and our liver, which are the kind of three places that we have sinusoids, not a whole bunch of places. Picture on this page doesn't show a sinusoid anywhere because they're not very commonly found capillaries. So as it reads, most plasma proteins, red blood cells cannot cross except in sinusoids, which have intercellular clefts so large that proteins and blood cells leave through their walls, and it could also be entering because it's not necessarily leaving. Proteins synthesized in our liver by way of cells called hepatocytes in our bloodstream to become plasma proteins like albumin, gamma globulins, fibrinogen, all those things that we talked about in our blood lecture a few weeks ago. And then blood cells enter our blood by way of our red bone marrow, as I mentioned. The next two ways that we move stuff in and out of our blood are listed here on this page. It includes a process we actually described at the very beginning of the semester in our cell lecture called transcytosis. And then there's also what we call bulk flow, which is what that figure on the right in a very complicated picture is all about. Transcytosis is a process in which a small quantity of material in our blood plasma becomes enclosed with a pinocytic vesicle, what we're showing right here, where a substance is coming into the cell, and by way of endocytosis, we form a vesicle. And then we move that substance across the cell to the other side, where we then push it out through exocytosis. Okay. So remember, trans means across or through. So in other words, we're pulling a substance through the cell and out of the blood, first by endo and then second by exocytosis. This is what we do for large lipid-insoluble molecules that cannot cross the capillary walls any other way, like insulin, which is how it enters our bloodstream, not what we're showing here on this page. Finally, bulk flow is the one that we will talk about here, where we move a lot of stuff at the same time, which is why it's called bulk flow. If you go to Costco or Sam's Club, you buy in bulk, you're buying a bunch of stuff, right? So bulk flow is a flow of a bunch of stuff. It's a passive process, so no ATP is needed, in which we we move large numbers of ions, molecules, or particles in a fluid, moving them together in the same direction. It is based on pressure gradients from areas of high pressure to low pressure. It is how we help regulate relative volumes of blood in interstitial fluid. This is something we'll talk a lot more about when we get to our urinary system lecture, which is the lecture where we talk about forming urine. And by forming urine, we're pulling water out of the blood along with other stuff. And in doing so, we're regulating how much blood we have, the volume of our blood, as I said. Okay. And that also helps regulate interstitial fluid volumes. Okay. It all relates to 4 major pressures that can be basically put together, and it's a calculation that comes up with a sum, or how do I want to say this? It comes out as what we call net filtration pressure. It's a balance of these different pressures. Some of them we can see representing with the arrows point into the blood. Some of them are pointing away from the blood, like these here, meaning we're trying to pull stuff in or out of the blood. And there's 4 pressures: BHP, IFHP, BCOP, IFOP, and then you calculate them up by subtracting and ultimately getting this net filtration pressure, where it's either going to be a positive number or a negative number. If the number is positive, it promotes what we call filtration. Pressure-driven movement of fluid and solutes from our blood capillaries into interstitial fluid. So filtration, in other words, means stuff leaves the blood. Let me say that again, kind of simplify it. Filtration means stuff leaves the blood. Whereas reabsorption, think of it simply as stuff enters the blood. Or actually, a better way to say it is even returns to the blood. So filtration, stuff leaves the blood. Reabsorption, stuff returns to the blood. Now, I said just a little bit ago, we're going to talk a lot more about this when we get to our urinary system lecture. There are 3 important processes that go into forming urine that take place in our kidneys. And first is called glomerular filtration, where we're pulling stuff out of the blood, performing filtration, as it states. Then we're performing what's called tubular reabsorption, which is where we return a lot of that stuff to the blood. Or we also perform the third tubular secretion, which is where we're moving it back out of the blood into the tubes. We'll get into all that stuff later on the semester. So, positive number NFP tells you that we're going to promote filtration, stuff leaving the blood. Okay. Reabsorption, stuff returns to the blood, and that's a negative NFP. I'm not going to ask you to know how to do the calculation or anything like that, but I do want you to know what a positive NFP means, what a negative NFP means, and what the term filtration means, what the term reabsorption means. This is why I kind of broke it down into simple terms. Okay, let's talk blood flow again. Now, thinking back to the heart lecture, like I said, hopefully everybody got a chance to watch. We talked, and there was a title slide that said, how does blood flow, or how does it move? And it said in parentheses, part 1. I don't know why, I must have deleted it a long time ago and I need to add it back in. But this is part 2, where we're talking about blood flowing through our vessels. which still is determined like it is in our heart with pressure gradients from high pressure to low pressure. But in our circulation, where blood flows through a tube, in other words, through vessels, there's an additional factor that can influence the flow of blood. We call this resistance. If you resist something, what does that mean? Do you want it to happen or do you not want it to happen? You don't. You're opposing it, right? I use the word oppose because you see the term there, and I used it in the sentence opposition. So when a fluid is moving through a tube, the opposition to its movement is called resistance. In our cardiovascular system, opposition to blood flow in a vessel is due to friction between the blood and the walls of our blood vessels, and we call this vascular resistance. Sometimes you'll see it listed as systemic vascular resistance. That's a measure of all resistances. And there's three factors that affect resistance. Okay, the size of the blood vessel lumen. If I go back to this picture, the lumen is the space where the blood flows. Okay, just looking at arteries and capillaries, which of those is going to resist the flow of blood more? An artery or an artery or a vein? Why do you say that? Look at the size of the lumen. It's harder for blood to flow through a smaller artery than it would be for a vein, because the veins are more wide open, right? Just like if you kink a garden hose, it's harder for the flow of water to go through it. The viscosity of blood. We talked about viscosity of blood. Back in our blood lecture, we said it's a little bit thicker than water. Does that change very easily, though? How thick our blood is? Can we change viscosity really easily? The answer to that is no. When I say easily, I mean like from right now to 10 seconds from now. No. But we can perform constriction and dilation pretty easily, can't we? We can decrease the size of our blood vessel, or we can increase the size of the blood vessel lumen. What about the total blood vessel length? How long our blood vessels are? Can we change that in 10 seconds very easily? No. It's not like we can grow our blood vessels longer or shorter in a short amount of time. That takes a long period of time for that to take place. So the lumen is the biggest factor, which, if you look at the bottom of the page here, we calculate resistance as 8 times viscosity times tube length divided by, that's a terrible symbol there, but that's pi, 3.14 da 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 da, times the radius. The radius, in other words, refers to the blood vessel's lumen to the 4th power. Now, by putting it to the 4th power, you're not multiplying it times 4, you're multiplying it times itself 4 times, right? Which means you change the lumen just a little bit, you're going to see huge changes to resistance. Now, I'm not going to have you calculate anything on the test, I promise you that. Which includes these figures, or these, not figures, but these at the bottom of this page. So, since blood viscosity doesn't change in healthy individuals, nor does the length of their blood vessels, the only method the cardiovascular system has to regulate vascular resistance is through changes in their vessel diameter. Notice that since vessel radius, since the vessel radius is to the 4th power, going back to this page, a small change in the radius or a small change in the lumen's diameter, because radius is half of the diameter, it's halfway across, translates to a very large change in resistance. In other words, the smaller the lumen of a blood vessel, the greater its resistance to blood flow. Going back to this slide, I said that arterioles are described as resistance vessels because they have the biggest opposition to flow. And they're the ones that most regulate our vascular resistance because they're the ones that can really perform lots of constriction dilation. Now, blood viscosity can change. Like if you're dehydrated and you have less water, there's less water in your blood, so your blood becomes a little bit thicker. Or by polycythemia, like I talked about blood doping, how you can inject more red blood cells in your body, that makes your blood a little thicker, increasing viscosity. Your blood's a little more like sludge pumping through your veins and arteries. Anemia, which remember is not necessarily low iron, but like, lack of red blood cells, or hemorrhage when you are bleeding, those decrease viscosity. So that will affect resistance. Obese people often have hypertension. What's hypertension? High blood pressure. Because they've got a lot of adipose tissue and fat needs blood vessels. So that increases the length of their blood vessels. So that affects resistance, and resistance and blood pressure go hand in hand. We can describe the relationship between blood flow as an inverse relationship to resistance. When I have an inverse relationship, what does that mean? If one goes up, what does the other do? Goes down. So write this down. Any increase in vascular resistance does what to blood flow? Decrease. One goes up, the other one goes down. Vascular resistance goes up, blood flow goes down. If we flip it and say vascular resistance goes down, what happens to blood flow? Increases. Let me say it again. Vascular resistance goes up, blood flow goes down. Vascular resistance goes down, blood flow goes up. And that's because vascular resistance here on this page is always in the denominator. It's on the opposite side, what blood flow would be. So changes here produce opposite changes here. Another term for systemic vascular resistance is total peripheral resistance. Abbreviations for those are SVR, which you see here a couple different times. SVR is short for systemic vascular resistance. You also see TPR, total peripheral resistance. It's just all vascular resistances offered by systemic blood vessels. The smaller the vessel, the more resistance it has. So your smallest vessels, your capillaries, your venules, your arterioles, offer the most SVR. I told you that here a little bit ago. A major function of our arterioles is to control SVR, which thus makes them responsible for controlling BP. What's BP short for? Blood pressure. And blood flow by performing vasodilation, vasoconstriction. The main center for regulating SVR is the vasomotor center in our brain stem, specifically the medulla oblongata of our brain stem. Let's look at the figure at the bottom here. We have blood vessel A, blood vessel B, blood vessel C. Obviously, A is the biggest, C is the smallest, right? Didn't take a genius to know that. We aren't going to give any specific units of measurement here. We'll just say the radius here for vessel A is 1, 1/2, and 1/4. So in other words, this is 4 times smaller than this one. Okay. Resistance, through its various calculation, for A is also 1. Blood vessel B, even though it's 1/2 as big as A, its resistance is 16 times greater. Look at the resistance here. 256 times that of A. And that's because, once again, it's to that fourth power. So a small change in lumen radius produces a big change overall. And as I said, resistance goes up. What happens to blood flow? Goes down. Blood flow through a small blood vessel like blood vessel C is much less than bigger blood vessels. Hopefully that's pretty common sense, right? Now, if vascular resistance is high, the only way to maintain blood flow is by increasing a pressure gradient, which is done by making our heart generate more pressure with each contraction. In other words, if blood pressure is high, vascular resistance is high, what's that mean your heart has to do? Pump harder. And I talked a little bit about that in that last lecture. Going back to that lecture on the heart, I told you guys how many times our heart beats a day, how many times it beats a year, how many times it beats in a lifetime, 2.5 billion or something like that. What we don't want is our heart working any harder than it needs to. We want it to, beat without much resistance. What I'm getting at is high blood pressure is very dangerous. It wears your heart out over time. Constantly having to beat hard to pump that blood out if you have resistance elevated. And guess what causes high resistance? Things like atherosclerosis, plaque in the arteries, coronary artery disease, especially specific to the heart. We'll talk a little bit about those later in the lecture. Speaking of blood pressure, once again, abbreviated BP. Thinking back to our last lecture, we learned that when you get your blood pressure, and this is not something you probably learned in that lecture, but when you go to the doctor and you get your blood pressure taken, they give you two numbers, right? That's because there's two different pressures in your vessels. One when your heart is beating, specifically your ventricles. That's the higher number. We call that the systolic number because that's when your heart is in systole. Systole is a fancy word for contraction. The lower number is called the diastolic number, which is the number when your heart is in a resting state, in diastole. So contracting your ventricles generates blood pressure. The fancy definition of blood pressure is the hydrostatic pressure exerted by our blood on the walls of a blood vessel. Several factors affect blood pressure. Cardiac output, a thing that we learned in our last lecture. Cardiac output, remember, is a factor of heart rate times stroke volume. So how fast your heart beats and how much blood it's pumping each time affects blood pressure. Total blood volume. Remember, total blood volume refers to how much blood you have, right? What do you think high blood volume is going to do to your blood pressure? Any increase in blood volume means more blood, which means higher blood pressure. And then vascular resistance, as we were just talking a lot about. Now, remember, we've been talking about it today, and we talked about it in the heart lecture. The flow of blood relies on pressure gradients, right? Well, blood pressure has to progressively drop the farther you get from the heart. Now, I don't mean like physical measurable distance. I mean distance traveled. Because like your superior and your inferior vena cava are basically right next to your left ventricle, right? They're attached to your right atrium, but they're right next, very close by your left ventricle where we're originally pumping blood out. But your superior and your inferior vena cavas are the last place blood flows as it returns back to the heart. So that's the farthest down the line. Look at the pressure of blood as it approaches our vena cavas. It's high, especially in our arteries. A lot of that has to do with those thick walls and the size of the lumen that we talked about earlier. And it drops progressively, pretty substantially, really, in the capillaries, and then continuously through the veins. And this helps create the pressure gradient, not to flow blood out of the heart, but to return it to the heart. We want blood flow to come back to the heart, right? Which means that we need pressure gradients that establish it back to the heart. Now, if I go back to this page, we said blood flow is inversely related to vascular resistance. I didn't really mention the top of the calculation, pressure 1 minus pressure 2, or it can be written as mean arterial pressure. In math class, I'm not asking you to do it, but if you calculate a mean, what are you calculating? Average. Average. So instead of having two numbers, like 120 over 80, you can actually combine them and get the average blood pressure in your vessels. We call this mean arterial pressure. Now, it's not as simple as just adding them together and dividing by 2, because if it was 120 over 80, you add those up together, that's 200. You divide by 2, that's 100, right? Well, mean arterial pressure of somebody that's 120 over 80 is more like 93-ish, if I'm thinking correctly off the top of my head, which is not perfectly right between 120 and 80, is it? That's because we don't spend equal time in systole and diastole. At rest, about a third of the time, your heart is contracting. The other 2/3 of the time, your heart is relaxed, which is why you can see the calculation is diastolic BP plus 1/3 systolic minus diastolic. Not something I'm going to ask you to know. Not something I'm going to ask you to calculate. It's kind of food for your thought. Great thing is, you know, if you guys are going into nursing or whatever it is, a lot of the blood pressure stuff is done pretty much easily with electronics. Okay? There's still going to be times where you're going to have to do it the old-fashioned way, but you see these a lot. Like you can go to Walmart and get your blood pressure taken, sitting there while you're waiting on your pharmacy order or whatever. And a lot of them calculate mean arterial pressure right off the bat easily as well. So kind of handy. Figure here on this next slide is just showing you all the factors that affect mean arterial pressure, in other words, blood pressure. Okay. Two major things are cardiac output and systemic vascular resistance. Cardiac output, we learned, is a factor of heart rate times stroke volume. What we learned last time, not last time because I recorded it, but in our last lecture, how you can increase your heart rate. Things like increasing sympathetic activity, epinephrine when we have the fight or flight response active. Epinephrine and norepinephrine, remember, are hormones from the adrenal medulla. Or by just decreasing activity of the parasympathetic nervous system. Stroke volume, amount of blood that our heart pumps each time it beats. We learned that contractility, preload, afterload, those all affect stroke volume. Stroke volume is really, really related to venous return, which we'll be getting into in just one second. And then we just talked about in this lecture vascular resistance, 3 different ways to affect it: viscosity of our blood, the vessel lumen diameter or radius, as it's listed here, and then the other being total blood vessel length. Now we're all showing here in this figure how to increase mean arterial pressure. We could flip it and say decrease mean arterial pressure, which means we do the opposite of what's listed here. You know, for like decreasing systemic vascular resistance, that's going to decrease blood pressure, so on and so forth. I snagged this figure from a different textbook, but it's kind of handy. It's got a lot of information in it. Speaking of venous return, I said we'd talk about that here in a second. Here is that second. Probably our last thing to talk about. And it's exactly what it sounds like. The volume of blood that's flowing back to our heart by way of our veins. In other words, the return of blood to our heart, which occurs due to pressure generated by contracting our left ventricle, which establishes that gradient. And then we drop that pressure progressively all the way back to our heart. Once we get that blood into our veins, there's not a big change in pressure. In our venules, it's about 16 millimeters of mercury. Our right ventricle is about 0. That's still sufficient enough to promote venous return. Now, back in our lymphatic lecture, we talked about the muscle pump and how that helps promote the movement of lymph. The same thing exists here with the return of blood from places like our legs that we show here. And the fact that we've got valves in our veins, I talked about those earlier, help prevent blood from flowing back when our blood moves forward. It's only going to go in one direction. We also talked about the respiratory pump, using pressure changes in our thoracic and abdominal cavities when we breathe, dropping pressure in our chest, below pressure in our abdomen, helps bring that blood up, or in the case of lymph, up towards our chest, towards our heart. And then something I mentioned, not a super promoter of this, but it does help venoconstriction, reducing the volume of blood stored in our veins to return it back to the heart. So all those different things discussed here promote venous return. Okay, we'll stop here because it's 10:51. We will finish this up on Monday. Yes, I won't record the rest of it. We'll just do it in class and then go from there. Have a fantastic weekend. I will see you all on Monday.